Cancer vaccines are like Hollywood wartime spies. Some offer an elegant subterfuge, effectively undermining a tumour from the inside. Others are more like Ernest Hemingway, larger than life, expensive, and not all that useful to their Russian handler. I'm Rachel Williamson, and this is Phase 3. Cancer vaccines started as prevention. First, there was the vaccine for TB. It is still used as a frontline therapy for bladder cancer today. Then in the 1980s, you had the hepatitis B vaccine. Stop Hep B and you can prevent the side effect of liver cancer. Finally, there's Gardasil, the revelatory vaccine that prevents human papillomavirus, or HPV. It has reduced cervical cancer rates by 60 to 90%, and it's saved thousands of women's lives since launching in 2006. But these are all cancers that are caused by viruses. In this series, we will investigate the new frontier, cancer vaccines as therapy. That is, when the cancer has already started. Therapy vaccines don't vaccinate the body against something. Instead, they show the immune system what a tumour is by highlighting the neoantigens on its surface. A neoantigen is a mutant protein that is unique to the tumour. Doing that turns the formerly hidden tumour cell into a glowing target. Add a drug that ramps up the immune system and the body can get to work. Merck and Moderna are so close to getting an mRNA vaccine for melanoma to work. It's in Phase 3 clinical trials. Elicio Therapeutics has a peptide vaccine for pancreatic cancer in Phase 2 trials. Bavarian Nordic is a bit different, using a virus to light up prostate tumours and to deliver a toxic payload. It's in Phase 3 trials. These are just a handful of the still small number of clinical trials and research going into cancer vaccines today. But to look forward, sometimes we need to look back first. Back to 1989, where Dr Ian Frazer met virologist Dr Jian Zhou and started on the path to making Gardasil. Dr Zhou died in 1999, but Ian is still actively advising cancer vaccine hopefuls. I'm very excited to talk about your history and your work because it has affected all the women I know, really. You famously co-invented the human papillomavirus, or HPV, vaccine. Now, this tricks the immune system into recognizing the real thing using what's called a virus-like particle, or VLP. Where did the VLP concept originally come from?
Oh, we have to go back quite a bit in time for that. I had to Professor Harold Zurhausen, who was working in the 1940s and 1950s on the connection between papillomavirus infection and cancer. That led to a lot of work mapping out, first of all, that there was a unique papillomavirus which was associated with cancers in humans, HPV 16. We now know there are several other ones, but 16 was the dominant one and still is. And then people started to work out how perhaps the virus could cause cancer. And that turned out to be due to the virus infecting cells and not producing more virus, but rather changing the program of the cells so that they became able to grow indefinitely and then perhaps become cancerous.
Seems like a simple fix, right? Stop HPV, stop cervical cancer. But here comes challenge number 1. You can't grow an HPV virus in the lab. Why not? Because the only place to get material is from cervical cancer patients, and cancer cells don't contain the whole HPV virus genome, which meant the usual experiments a virologist could do were off the table. But Ian and Jean found a workaround.
We knew the virus was basically coated with two particular proteins, which we called L1 and L2 for no particularly good reason. And those two proteins packaged up the infectious material, the genetic information of the virus. So we started thinking about, could we make a virus in the lab by expressing L1 and L2 proteins using the then new technologies of genetic engineering. And that sounded easy in principle, but in practice turned out to be quite hard.
And here is challenge number 2. They struggled to find a complete L1 or L2 genome. Why? Because the material they had to work with was from cancer. And what is cancer notorious for? Changing genetic information. John and Ian started their work in the UK before moving to Australia, but it was Australia where they got lucky with a sample that contained the real code for L1. It took a year, but they were able to build a complete gene for both proteins.
Much to our surprise, if we expressed these genes in the test tube in the right way, we found these things called virus-like particles. And these virus-like particles were not virus. They were empty. There was nothing infectious about them. But they looked like the virus as far as the body's immune system was concerned.
What made the HPV virus so amenable to creating a virus-like particle? Because a lot of other companies have tried this for cancer and gotten absolutely nowhere.
Well, you have to understand, of course, that the vast majorities of cancers are not triggered off by infection. You know, the basic message in cancer is that the genetic information of the host cell that's going to become a cancer is altered. And it can be altered by radiation, it can be altered by drugs, it can be altered by virus. But there are really only a few cancers that are caused by virus infections. And the virus causes cancer not using the capsid, not using the outside. It uses the genetic information inside the virus to change the nature of the cells so they become cancerous. So we have to basically stop the infection with the virus getting into cells in order to stop the cells becoming cancerous. So that was basically what we aimed to do, and what we were able to show. We could make an antibody that would basically stop the virus getting into cells. That took a bit of work, and once we had done it, other groups copied us, checked that we'd got it right. Some claimed they got there first, but in the long run, it turned out that we were probably first.
The patent battle over this blockbuster drug was intense. Several groups were working on the same concept around the same time. In 2005, a US court awarded the patent to a team from Georgetown University, saying Ian's and John's work was inaccurate. 2 years later, that was reversed on appeal. It's been a ride. But preventative vaccines are also a form whose time has kind of passed, at least in terms of cutting-edge science. Using vaccines to infect tumours, therapeutic vaccines, that is where the science is at. After Gardasil, Ian's next step was into treating the cancer caused by HPV. 25 years on, that's still the goal, and not just for him. Pairing a vaccine to light up cancer cells with a drug that turns up the volume on the body's immune system is the dream. One of these paired immunotherapies are monoclonal antibodies. These are lab-made proteins that mimic the antibodies produced naturally by the immune system. But herein lies another challenge. I'll let Ian explain.
Of course, there are complications from that as well. If you turn up the immune system, your immune system doesn't really distinguish well between cancer and normal cells, and therefore there's always a risk that there will be side effects caused by too much immunity. We're in an interesting situation at the moment that we're learning very rapidly that an idea which was had a long time ago, that the body's own defenses against infection also control, to some extent, against cancer, is probably true. In other words, we have an immune system which is really there to prevent us getting infected with viruses and bacteria, but the immune system can also recognize a tumor if it's sufficiently different from ourselves. We know that there must be something really significant there, because if you lack an immune system, you become more prone to cancer. And so that gives us confidence that we can use the immune system. We can immunize patients with something that the tumor might have that the body hasn't had a chance to fight.
Again, this sounds simple, but in fact it's really hard. Because if you remember, cancer cells change. When it comes to the work Ian is doing with another company, which is working on a vaccine to treat cancer caused by HPV, to some extent every treatment has to be personalised to that patient's cancer. I asked Ian whether that means an allergenic or off-the-shelf vaccine treatment for cervical cancer is possible.
Well, it's not quite as bad as that. I mean, what we're now beginning to realize is that those abnormal pathways include parts of the immune system, and we're trying to correct, if you like, the missing bits of the immune system to push the immune response along and try and help clear the cancer. So the idea is more complicated for a therapeutic vaccine than it is for a preventative vaccine, where we know there's a virus and we just want to make sure the virus doesn't get in and cause cancer.
Therapeutic vaccines seem to be having a moment right now. Which modality do you think is going to have the most legs?
Well, right at the moment, monoclonal antibody therapy definitely comes into that category for treating cancers when they can't just be cut out. We recognise that for some sorts of cancers, they can be curative, and for others, they can at least prevent the cancer spreading. In the future, there are other possibilities which are more experimental. mRNA, let's call it immunotherapies. I don't like the word vaccines for these things. mRNA is just one mode of turning on the immune system, programming the immune system. But mRNA can also be used as a specific means of targeting a cancer. I mean, it can kill cancer cells using non-immunological mechanisms. We're very much at the experimental stage of mRNA vaccines at the moment. Obviously, I think COVID has moved them along very much more rapidly than would otherwise have been the case. That means we're sort of catching up our scientific knowledge with the practical knowledge that these can be useful. And I think that the important thing of all of this is to remember that you invest in the science because it gives you a logical way of dealing with something rather than just an empirical way of doing it. And the more we understand about what we're trying to do, the more likely we are to be on target. And that's true whether we're talking about vaccines, whether we're talking about cancer treatments. It's always science that drives the steps forward which are logical rather than just trial and error.
That was Dr Ian Frazer, the coinventor of HPV vaccine, Gardasil. If building a vaccine from nothing sounds hard, building Trojan horses to infect tumours is too. But that's what the financiers are backing these days, as we'll find out after this break.
Hi there, I'm Karis Palmer, producer of Phase 3. When Rachel and I set about building a new podcast for life science leaders, scientists and long-suffering biotech investors, we looked at what was missing in this space. We believe Phase 3 serves an unmet need for in-depth conversations in a world where nuance matters and AI-written investment articles simply won't cut it. If you agree, please follow us and sign up to our newsletter via LinkedIn. Pledge financial support at phase3.buzzsprout.com and rate and review the podcast on the podcast platform you use to help bring it to the attention of others. Now back to the show.
Cancer vaccines will be a success story thanks to 3 pieces of technology: neoantigens, AI, and better ways to get vaccines inside the body. That and the startling rise of mRNA, of course. Wilson's advisory lead healthcare analyst, Dr Shane Storey, is fascinated by the elegance of vaccines. Instead of using a single toxic drug to hammer the body, engage the power latent within the body itself. So I had to ask him, what is so alluring about the vaccine concept for cancer?
Well, I've always loved it. I looked at it maybe 20 years ago. I've always been intrigued by it. I think it's an alluring idea because it gets us away from the single drug, single receptor, very simplistic kind of view that one interaction is going to solve all the things. And when you see the opportunity to, recruit the sophistication and just the power, sitting there in every cell in the immune system. The system is already doing half the job for you in that in a healthy individual, the immune system's constantly doing surveillance and checking for transformed cells it needs to get rid of. And it's got such a devastating range of weapons that if you're able to get it to work on your behalf, then you'd think that you would have a very good chance of being able to impact, the disease much more so than you can with like a single cytotoxic drug. So that's why it's always been such an appealing idea.
Are investors backing in the vaccine concept yet, given it hasn't proved itself to be particularly successful yet?
I think investors are backing in the RNA concept.
And not the cell-based or...
No, and what I mean by that, if you look at the last 5 years maybe, I think there's probably been, 3 things that have come through that have made vaccines for cancer doable again in most people's mind. Checkpoints is one of them. The other is ideally working side by side with checkpoints is something that's quite selective. And so those subtle differences between cancer cells and normal human cells, they're called neoantigens. And I think there's been a lot of advances in identifying which ones they are, which ones will work, ideally with the immune system that you're trying to get it to interact with.
Checkpoints and neoantigens. These are 2 really important elements of modern cancer treatment that come with a backstory. Checkpoint inhibitor therapy targets the immune system's built-in checkpoints. Basically, it targets proteins that act as brakes on the immune system. But it doesn't always work for patients whose immune system is already weakened, and it doesn't work for cold tumours which hide and don't kick off immune activity. These include pancreatic and ovarian cancers. One way to turn a secretive tumour into a bright red glowing hit me button is to infect it with a vaccine. Antigens trigger an immune response. Neoantigens are specific to tumours. They can be used to light up cold tumours, to make personalised cancer therapies, and even give T cells a memory of what the cancer looks like. The University of Oxford's LungVax clinical trial was launched this year, and it uses neoantigens to create targets for the immune system to attack. But these are not the most significant pieces of research to come out in the last decade. I'll let Shane explain.
Ultimately, possibly the most important bit, and one that was probably not foreseeable maybe even 10 years ago, was like the amazing rise of RNA as a modality. I think drug developers of my age would never have picked RNA to have won the war in COVID. We would have chosen something entirely different, and I think it's just because we're part of that traumatized generation who could just never work with RNA in a lab. Never thought it would ever get to a point where it's just been so miraculously industrialized. So if you look at the companies that are doing, the most interesting work now, and some of them even in combination with checkpoints, as we've been discussing, like BioNTech and Moderna, I think investors are backing in the RNA concept and vaccines more generally, but then increasingly looking at, well, it might also pull the cancer vaccine thing off too. So I think they're backing RNA in, and I think they'll get vaccines. They'll get vaccines for free. Right.
There was some early success with Gardasil, the very, very famous HPV vaccine, and that borrowed from infectious disease vaccines. What did that do for the growth and commercialization attempts of that early science?
That was a terrific vaccine, and it worked because Ian Frazer and Jianzhu, the inventors, they worked out that to get an immune response against human papillomavirus, all you had to come up with, I say all you had to, it was actually quite technically very difficult, and worse, even harder to manufacture it, was to create an empty virus particle. So a virus particle that had the external capsid, is what it's called, but had none of the guts, had none of the infectious, nucleic acid inside it. And of course, because that virus has a causative impact on cervical cancer and oral cancer and other cancers. By extension, you immunize people against HPV and you eradicate those forms of cancers. And that was, commercially an incredible success for Merck and for CSL and for UQ as well. But then that didn't always work. People then went after Epstein-Barr and a couple of other different viruses and found that, like many viruses, they're actually quite tricky to do.
What impact did that have? Did it spur a lot of companies and commercialization attempts for other ideas?
No, it definitely did. And it probably led a lot of vaccine developers down the wrong path. And by that I mean, if you look back around the time when Gardasil was successful, so the early 2000s, I think it was approved 2006. If you look around that era, cancer vaccines, viruses were part of the scene. They were either being used directly in the Gardasil way, or they were being used as vectors to try and create what were called vector-mediated vaccines, or even DNA vaccines. And they were the initial attempts. And I can't even imagine why we didn't think of RNA at the time, because you think of the complications of trying to get a DNA molecule inside a cell, inside the nucleus, not have it integrate in some problematic piece of the genome, hopefully get transcribed into RNA. And that it's just so complicated. When you just go in with an RNA and the thing just sits in the cytoplasm and just works and makes proteins. It just was obvious. But maybe Gardasil led people down the wrong path. And none of those vaccines worked.
Let's talk about one that has worked, Sipuleucel T, which is better known by its brand name Provenge. It treats prostate cancer. What has that taught us about how to build a therapeutic cancer vaccine, both from a science perspective, but also from building a company or a infrastructure to actually sell it, given the failures of the past?
Well, it was smart, right? So Provenge was really almost like in vitro immunization, in that they took the immune cells out of the patient, rubbed their little noses in the antigen, and then put the cells back in the patient. And because those cells had learned to go after a particular protein on prostate cancer, that was your vaccine, right? So it was a very clever idea, and it did work, and it achieved the benefit for the patients. In the end, though, it didn't work really as a product, because that manufacturing was so expensive, because every patient basically gets the bespoke little manufacturing process. And the same is true these days of CAR-T therapies, where the manufacturing costs are fairly prohibitive, which means the product price has to be so high that then the insurers don't like to pay it, and so it kind of all unravels. So brilliant scientifically, but I'd have to say probably failed as a product because of
Is that a direction that companies are looking at again today?
They still are, yeah. They're still looking at it, and they're getting even more sophisticated in what they do and how they make them. But there are still, I guess they're called dendritic cell vaccines. There's still a couple in development, not many. Between you and me, I just don't think they're going to win the war. I think RNA is going to win in this setting.
Now, after years in the desert, the big comeback for cancer vaccines, as we've mentioned, has been obviously mRNA, and what Moderna and Merck and BioNTech and a few others are doing. Much of it in Melbourne, from what I can see. What are these doing for the industry? How excited are people? What sort of money are they bringing in to back science? What are they building?
Look, it is really exciting time. And yes, you're right. A lot of it is being done here locally. And a lot of it is with RNA. But then it's not as simple these days. So go back to the idea of the antigens, which none of this is going to work without good antigens. There's science that's going on there where you can either, and this is really interesting, where you could either try and find an antigen that is made in a wide number of patients who have a particular cancer. And in that case, it allows you to sort of have an off-the-shelf sort of vaccine that you hope will protect a lot of people to varying degrees. Or you can now look for a whole range of personalized neoantigens that you get for an individual patient by sequencing that patient's tumor, and you can come up with some of those vaccine candidates now to come up with a very powerful personalized
It sounds very expensive.
vaccine. But it's not with RNA, see, because with RNA you can just dial up a change in the sequence, and then you can actually do that in a modular kind of way. So it's nowhere near as expensive as the dendritic cell thing we discussed before. That's really expensive. But you can dial that up pretty simply in a small scale batch manufacturing with RNA and do it that way. And there are trials that Moderna have. And I think BioNTech have got that personalized approach in some of their phase 3 programs. The other interesting thing is using AI so rather than sequence a patient's tumor, if you get the genetic information, use AI to sort of detect just from those sequences, which are likely to be good neoantigens and also likely to interact with the right bits of the immune system in the right way at the right time to do the right things. That's where you're starting to see a lot of really interesting research.
What companies are really exciting you right now?
We spoke about RNA as the key here, but there's still some drawbacks there that a number of companies are looking at, and it really is to do with how they're delivered. The vaccine industry is still absolutely over-dependent on, say, intramuscular injections, say. There's hardly any immune cells in a muscle. So I like companies like VAXXED who are doing away with needles, and they're using patch-based technologies to put antigen just underneath the surface of the skin where, surprise, surprise, all the antigen-presenting cells hang out, right? So you're putting the antigen in the right place, which is a terrific start. So we like that. We also like companies like Arcturus in the States who are doing RNA, but their RNAs are self-replicating. So they make copies of themselves inside the cell. So a sixth of the material is needed to generate a superior immune response because the vaccine has that self-replicating nature. So I guess the companies that we look for are those that are trying to solve the delivery issues. If we start from the assumption that RNA is probably going to win, what companies can we find that are going to help it win even more? I think they're the ones that interest us as analysts.
That was Dr Shane Storey, head of the healthcare team at Wilson's Advisory. Shane says mRNA cancer vaccines will be on the market in 5 years, and these will change the way the cancer immunotherapy industry works. They can be made cheaply enough to give in combination with an expensive checkpoint inhibitor. In contrast, a cell vaccine like Provenge is never going to work just on cost. In our next episode, we start deep diving into companies that are making cancer vaccines in Australia, but doing so just a little bit differently again.